March 6, 1937. Nature 399. The Ancestry of Insects. By Dr. A. D. Ims, F.R.S. The insects are unknown in their original or primitive manifestations, and consequently their ancestry is wrapped in obscurity. The subject can only be approached by the coordination of whatever evidence may be gleaned from three principal courts of appeal: comparative morphology, embryology, and paleontology. Of these, paleontology has, so far, remained almost like a closed book. No annectant forms bearing directly upon the problem have been disclosed in the strata of any geological period. Without this necessary record of the past, the links in the evolutionary chain are deductions based mainly upon morphology and development. For a considerable time past, the two most plausible theories of insect descent have been those which involved either the Crustacea or Symphyla as ancestors. The crustacean theory, which has based no external structure, is losing ground, while that involving the Symphyla has remained in much the same status that it acquired in the days of Packard. The most important evidence bearing upon insect ancestry that has come to hand during the last 30 years or more was Silvestri's one discovery, in 1903, of the remarkable Thesanoran Anogipix. Since that date, no revelation of comparable significance has been made. While this discovery added weight to the Symphylan theory, Packard's conclusion of 1898 too still holds good today. This writer stated that Scolopendrella is the only extant arthropod, which, with the sole exception of being progoniate, fulfills the conditions required an ancestor of the Thysanura and, through them, of the winged insects. In 1930, Tilliard III brought forward a theory which claimed that the insecta were derived from remote hypothetical nauplioid ancestors, Protaptera. They arose separately from the Symphyla and other Myriapoda which, he maintained, had a similar origin. The Symphylan theory was rejected by this author owing to the old crux of the segmental location of the gonopore. The contention of the present communication is that the Symphylan theory is substantiated by a larger body of evidence than any other explanation of insect descent. In considering this theory, it needs to be stressed that it is generally accepted. The present article is an abbreviation of one bearing the same title: Trans. Society. Brit. Entom. 1, 1936, the main features of which formed the subject of a presidential address given to the Society for British Entomology, at a congress held in Cambridge, OU, June 26–29, 1936. Today that among the Thysanura are found the most generalized of all insects. Lepisma and its allies are the forerunners of the Pterygoda, while Anogipix and Campodia are more nearly related to the ancestral insects. The old phylogenetic importance of Campodia has now been largely eclipsed by Anogipix with its greater display of Symphylan characters. Since Anogipix provides an essential link in the present theory, these characters need to be recounted. They include simple multi-articulate antennae, styli and eversible sacs on most of the abdominal segments, cerci bearing the orifices of large posterior glands, mouth parts built on an essentially similar plan, an identical leg segmentation with single-jointed tarsi, a simple digestive canal with homologous salivary glands and the same fundamental type of excretory organs. A second link in the chain is provided by embryology. The lower insects pass through a developmental phase revealing 14 postcephalic segments, each bearing limb rudiments. This phase can only be interpreted as a relic of a former polypodous condition. On turning to the Symphyla, it will be noted that they reveal, in their adult structure, close resemblance in metamerism as evidenced by the appendages, namely, 12 pairs of legs, the 13th pair transformed into sensory processes and the 14th pair, as in insects, represented by cerci. Thus it seems very possible that the insecta recapitulate what may be regarded as a Symphylan stage in their development. In the derivation of the insecta from Symphylan ancestors, it would appear that three main evolutionary changes were involved. L. The development of a segmental tracheal system. 2. Concentration of the locomotory function in the first three postcephalic segments, with the consequent reduction of the appendages of the remaining segments other than cerci to a vestigial condition. 3. The development of posterior gonoducts opening between the 10th and 11th postcephalic segments, together with the atrophy of the original ducts. L and 2 require little comment since they are evidenced in the development of the lower insects and in the adult structure of the Thysanura. In connection with 3, it is noteworthy that a shifting backwards of the genital apertures during development is a common feature among many orders of insects. Furthermore, new or secondary genital passages and Copyright 1937 Nature Publishing Group. 400 Nature March 6, 1937. Orifices have been acquired among Lepidoptera, Strepsiptera and in the Hemipterus family Simicydrae. The postulation of a change from a progoniate to an opistogoniate condition refers, therefore, to the evolution of a class wherein diverse modifications of the genital conduits are a prominent feature. The insecta are peculiar in that the egg follicles are disposed in a linear series forming ovarioles, while at the apex of each ovariole is an end chamber, or germarium containing the undifferentiated germ cells. In other arthropods no such arrangement into ovarioles prevails and the germarium is located parietally. It will be evident that structural modifications of the gonads must have been involved during the evolution of the insecta, and, it is claimed, that these changes occurred in association with the development of posterior gonoducts. So far, the Protura and Collembola have been omitted from discussion. They are side developments off the main line of insect descent. The Protura show a predominance of hexapod features, suggesting that the early evolution of this group was with the rest of the insecta. Afterwards they diverged as a specialized side branch but retained the primitive trait of anamorphosis. The Collembola display both myriapodan and insectan characters. Owing, possibly, to the paucity of yolk, which induced holoblastic segmentation, development became arrested and only 9 trunk segments formed. In the absence of ovarioles and retention of the parietal grammarium, together with the general simplicity of the gonads, the collembola betray close resemblances to the symphyla. The possibility that traces of a former progoniate ancestry are revealed in collembola, as mentioned by Claypole, needs further investigation. Evidence of such ancestry also requires exploration among other Apterogoda. Re-examination of the symphylan theory seems, therefore, to establish it as the only adequate explanation of insect descent. In view of the many fundamental similarities between the Symphyla and Tbisonura, it is claimed that these two groups arose from a common stock, not very different from living forms of Symphyla. Theories involving separate origins for the two groups in question, on the grounds of disparity in the segmental locations of the gonopores, have to fall back upon the improbable hypothesis of convergence to account for their community of structure. If the propositions now advanced serve to focus attention upon aspects of the problem hitherto neglected, they will have fulfilled their purpose. 1 Ann. Della Scuola Sup. D. Oric. In Portici. Roy. Sus. Tassum. 1930. The Recent Floods. By Dr. C. E. P. Brooks. The Recent Floods 1 and the United States. By Dr. C. E. P. Brooks. Teehee, Mississippi and its tributaries together drain about half the area of the United States. The Mississippi itself runs through the center of the country, the Missouri rises in the Rocky Mountains and the Ohio in the Allegheny Mountains. The upper courses of the Mississippi and Missouri lie in relatively dry regions, where moreover the winter precipitation is mainly in the form of snow. The Ohio, on the other hand, runs through a region of comparatively heavy rainfall, and is much more liable to flooding. During the last few months of 1936, the rainfall in the valley of the Ohio and the Mississippi above Memphis had been abnormally heavy, the total for the months September-December being nearly 1.5 times the normal. Economic importance of Blattioidea. E.g., cockroach. Economic importance. Cockroaches are among the most cosmopolitan of all insect pests. They are associated with human dwellings throughout the world, hiding in cracks and crevices during the day and emerging at night to forage for food and water. Although they do not sting or bite, they are usually associated with unsanitary conditions and may carry a variety of human pathogens on their bodies. The decomposing remains of dead roaches and their feces are an important source of household dander that becomes airborne and provokes respiratory allergies in sensitive people. But cockroaches also have proven to be very useful as research tools, particularly for the study of insect physiology and toxicology. Major families. Blattidae includes the oriental cockroach Blatta orientalis and the American cockroach Paraplaneta americana. Bladelidae includes the German cockroach Blattella germanica. Bug bites. The American cockroach, Paraplaneta americana, is an introduced species that probably originated in Africa. Some cockroaches in the family Polyphagidae live as commensals in the nests of ants. Gromphidorina portentosa, a species from Madagascar, can force air out of its tracheal system to produce an audible hiss. This behavior is used to scare predators. These large, slow-moving cockroaches are sometimes kept as pets. Despite their bad reputation, only about a dozen species, out of 4,000 worldwide, are regarded as pests. The Serenum cockroach, Pycnoscelis serenamensis, is the intermediate host of a nematode, Oxysporum mansoni, that may cause blindness in poultry. Hemiptera has two suborders, Heteroptera and Homoptera. Suborder Heteroptera. Wings overlapping on the abdomen, forewings are apically membranous, large pronotum, tarsi 3-jointed, metamorphosis incomplete. 2 series. 1. Gymnocerata, antenna conspicuous, freely movable in front of the head. 2. Cryptocerata, antenna concealed beneath the head, aquatic. Suborder Homoptera. Wings sloping along the sides of the body, forewings of uniform consistency, pronotum small, tarsi 1 to 3-jointed, metamorphosis incomplete. 2 series. 1. Ochinorentia, antennae very short, rostrum plainly arising from the head, tarsi 3-jointed, active forms. 2. Sternurentia, antennae well developed, rostrum arising between anterior coxae or wanting, tarsi 1 to 2-jointed, inactive forms. Heteroptera, Gymnocerata. 1. Family Pentatoidae, shield bugs. Largest family in Hemiptera. Scutellum very large, often covering the abdomen. Most are brilliantly colored with a nauseous odor due to the secretion from 2 ventral openings on either side of the metasternum. Vast majority are vegetable feeders, but some are predaceous on Lepidopteran larvae. Antennae 4 or 5 segmented, basally concealed by the lateral margin of the head. Tarsi 2 or 3 segmented, claws with pulvily adhesive pads. Hemelytra well developed and membranes have 5 to 12 veins. Eggs are barrel-shaped, laid in compact masses. Nymphs are flattened and rounded, brilliantly colored. Both sexes have stridulation property and able to eject an obnoxious fluid to a distance of 6 to 12 inches. Certain genera show sexual dimorphism, males forming horn-like projections. Only few are major plant pests. 2. Family Regiviidae. A large number of the members are predaceous, sometimes known as assassin bugs. Usually they feed on the blood of other insects but rarely attack other animals including human. They have long head LRGE eyes and ocelli. Antennae are long and geniculate with up to 40 segments. Rostrum 3 segmented and pointed. They are brown or black colored. Forelegs have adhesive pads on tibi. Predatory Regiviidae use the long rostrum to inject a lethal saliva that liquefies the insides of the prey, which are then sucked out. The saliva contains enzymes that digest the tissues they swallow. This process is generally referred to as extraintestinal digestion. The saliva is commonly effective at killing prey substantially larger than the bug itself. Some species are bloodsuckers rather than predators. Triatoma species and other members of the subfamily Triatominae, such as Rodnius species, are known as kissing bugs, because they tend to bite sleeping humans in the soft tissue around the lips and eyes. The punctures of species of Triatoma cause a burning pain lasting for 2 or 4 days, which is caused by the anticoagulin from the salivary gland. Rodnius prolixus of Central and South America transmit the potentially fatal trypanosomal Chagas, American trypanosomiasis disease, by acting as a vector of trypanosomal cruzi. Indirect transmission occurs when people scratch the skin, introducing feces into the bite wound. Other ways of inoculation are via the mucous membranes, eyes, for example blood transfusion, organ donation and food contamination. The saliva of the species Rhinocorus marginatus has some activity against human pathogenic gram-negative bacteria, Escherichia coli, Pseudomonas aeruginosa, Proteus vulgaris, and Salmonella typhimurium, and the gram-positive Streptococcus pyogenes. Reduvius personatus visits houses to feed on Cymex. Platymerus labicalis preys on coconut beetle and Rhinocorus fuscipes on the larva of Spodoptera. Reduvius personatus known as the masked hunter because of its habit of camouflaging itself with dust. Some species resemble other insects. E.g., Rapidosoma resembles Phasmidae. 3. Family Gerridae, pond skaters. Large slender body with elongate head, large eyes. Some species are apteris, hemelytra indifferentiated and reduced. Rostrum 4 segmented. Metathorax has a scent gland. Coxae rotatory, mid and hind legs elongate. Tarsi 2 segmented. Forelegs are raptorial with hydrofuge hair pile. Water surface dwellers and predaceous. Some species can be used for pest control. R.G. Geris tristan in freshwater ponds and paddy fields feed upon Niliparvata higgins. Heteroptera, Cryptocerida family Nepidae, water scorpion. Antennae 3 segmented, forelegs raptorial, prehensile hind legs notatory with hair fringe on tibia, tarsi 1 segmented. Abdomen has apical respiratory tube consisting of 2 elongate spine-like processes, each of which is grooved to form a half canal. Both halves are movable and locked together by hook-like bristles. The respiratory tube obtains air from the atmosphere and transfers to a pair of spiracles at its base. 3 pairs of false spiracles are present on the 3rd, 4th and 5th abdominal segments. They are covered by a membrane and function as a sieve. They also have sensory setae. Female has toothed ovipositor and the eggs are laid in the notches made on the petioles of aquatic plants. Eggs have apical filaments. Both nymphs and adults can stridulate. Homoptera, Ochinorintia. 1. Family Cicadidae. Large insects with entirely membranous wings. 3 subcontiguous ocelli, anterior femur thickened and spined beneath. Males with a sound-producing apparatus on either side at the base of the abdomen. One of the well-known species is the periodical cicada, which appears in large numbers after long periods as their nymphs take 13 to 17 years for development. Female deposits eggs in slits made on tree twigs. Eggs hatch in 6 weeks. Nymphs fall onto the ground and start leading a subterranean life, sucking juices from fine roots of trees. Under high temperature, the final stage nymphs construct cones of 4 inches high using earthen particles in which they live their final stage, then they remove their exuvia and come out as adults. Sound-producing mechanisms. Cicadas are best known for the stridulations of the male. Special organs called tymbals, which are membranes that vibrate very quickly when pulled by tiny muscles. Some types of cicadas have exposed tymbals, while some species have hidden tymbals and flex their abdomen to open their tymbal covers to modulate their sound. Each type of sound produced has a different meaning. Alarm, distress calls, pre-calls, calls to attract mates and establish a territory, courting calls and choruses. Some species of cicadas lack tymbals. They use their wings to make crackling or popping noises known as crepitation. Some cicadas possess rasp-like parts of their bodies which when stroked with part of a wing produces yet another type of cicada sound. 2. Family Cercopidae, frog hoppers, cuckoo spit insects. Ocelli 2 or none, antennal flagellum composed of large subpiriform base and a slender seta, posterior coxae short and conical, tibi cylindrical and armed with spines. The nymphs of some genera get enveloped in a frothy discharge called the cuckoo spit, which protects them from predators as well as from the sun. The tergites and plurites of the 3rd to 9th abdominal segments curve beneath the abdomen as membranous extensions that meet the midventral line, forming a cavity into which the spiracles open. The cavity is closed anteriorly, but air circulation is possible by a posterior valve. When the fluid from the anus reaches this chamber, it is blown into bubbles by the air expelled from the chamber through the valve. This forms the frothy discharge. On the 7th and 8th abdominal segments are lateral glands which produce a waxy substance that mixes with the frothy discharge, which gives it a soap-like consistency. 3. Family Membracidae, tree hoppers. Pronotum generally prolonged backward into a process which lies like a hood over the abdomen. Flagellum with many joints. Posterior coxae transverse. Tibi angular. Eggs are laid in small groups in two parallel slits made in the twigs of trees. In nymphs, the pronodal process is either absent or partially developed. Both nymphs and adults are commonly attended by ants, the ants stroking the body of the membracidae with antennae to feed on an anal secretion. Certain genera are affected by parasites causing castration parasitaire, where the external genitalia are reduced or modified. 4. Family Jacidae, Cicadelidae leaf hoppers. Acelli 2. Variable in position. Antenna has numerous joints. Pronotum never produced backwards. Posterior coxae transverse. Tibi angular in the hind pair with bristles or spines. The body is slender tapering posteriorly. They rest in a ready-to-jump position. When disturbed, they leap several feet and take to their wing. Eggs are elongated and laid in longitudinal rows on stems or on or beneath leaves. Nymphs pass through 6 instars with wing rudiments becoming prominent by 3rd instar. They attack cereal and fodder crops as well as fruit and forest trees. Series Sternurentia. Family Aphididae, plant lice. Only males are winged and both wings are of similar consistency. Tarsi 2-jointed, rostrum long and antennae 3 to 6-jointed. 5th abdominal segment is usually with a pair of dorsal cornicles, honey tubes, which secrete a waxy substance that helps to protect from predators. Many aphids also produce a white waxy substance from the dermal glands either in the form of a powder or as threads. They pass their life on young shoots and foliage of plants. Some live on roots and some others are gall formers. Natural enemies of aphids are Hymenoptera, Coccinellidae, Syrphidae. Aphids form one of the major pests of many cultivated crops like cotton, beans, banana, corn, wheat, coconut, etc. Various phenomena are there for reproduction. 1. Parthenogenesis. 2. Oviparity and viviparity. 3. Occurrence of generations in which sexes are unequally represented, males rare or even absent. They show high rate of fecundity, reproduction dependent on temperature and quality of host plant. Aphids are known for the polymorphism shown in different generations of the same species. 1. Different forms of the same sex either in the same or different generations. 2. Individuals with perfect or atrophied mouth parts. 3. Individuals of the same sex but difference in gonads. They show difference in their habits. 2. 1. Migration to different host plants. 2. Different modes of life of the same species on the same host plant. 3. Different habits of the individuals of the same generation. Life cycle. The following stages can be seen in the life history of an aphid. Eggs are laid by sexual oviparous females in the autumn. The egg stage continues throughout the winter. In the following spring the eggs hatch to form apteris, parthenogenetic viviparous females. These females give rise to many generations throughout summer of similar females on the primary host plants. Some of the progeny can be winged parthenogenetic females. The winged viviparous females migrate as swarms to secondary host plants and produce similar females. Towards the end of summer or autumn both apteris and parthenogenetic generations give rise to sexual males and females. The females are oviparous. The cycle repeats. Polymorphs in the life cycle of a migratory aphid. 1. Fundatrices, apteris, viviparous, parthenogenetic females which emerge in spring from the overwintering eggs. Sense organs, antennae and legs are not well developed, eyes are small. 2. Fundatrigenii, apteris, viviparous, parthenogenetic females which are progenies of fundatrices and live on the primary host. 3. Migrants, winged, viviparous, parthenogenetic females which develop on primary host and migrate to secondary host. 4. Alienicoli, winged and parthenogenetic viviparous females developing on the secondary host. 5. Sexuperae, winged parthenogenetic viviparous females developing on the secondary host migrate to the primary host. 6. Sexuales, form only once in the life cycle. Consists of sexual oviparous females and males which reproduce sexually to give rise to the next generation of fundatrices. Eggs winter apteris. Parthenogenetic. Viviparous. Fundatrices summer. Apteris. Parthenogenetic, viviparous. Fundatrigenii summer, primary host. Winged. Parthenogenetic, viviparous. Migrants summer, secondary host. Winged. Parthenogenetic, viviparous. Alienicoli summer, secondary host. Winged. Parthenogenetic, viviparous. Sexuperae end of summer, primary host. Sexual males and females, females oviparous. Sexuales winter, primary host, eggs give rise to fundatrices. Hemiptera has 2. Series Sternurentia. Family Aphididae, plant lice. Only males are winged and both wings are of similar consistency. Tarsi 2-jointed, rostrum long and antennae 3 to 6-jointed. 5th abdominal segment is usually with a pair of dorsal cornicles, honey tubes, which secrete a waxy substance that helps to protect from predators. Many aphids also produce a white waxy substance from the dermal glands either in the form of a powder or as threads. They pass their life on young shoots and foliage of plants. Some live on roots and some others are gall formers. Natural enemies of aphids are Hymenoptera, Coccinellidae, Syrphidae. Aphids form one of the major pests of many cultivated crops like cotton, beans, banana, corn, wheat, coconut, etc. Various phenomena are there for reproduction. 1. Parthenogenesis. 2. Oviparity and viviparity. 3. Occurrence of generations in which sexes are unequally represented, males rare or even absent. They show high rate of fecundity, reproduction dependent on temperature and quality of host plant. Aphids are known for the polymorphism shown in different generations of the same species. 1. Different forms of the same sex either in the same or different generations. 2. Individuals with perfect or atrophied mouth parts. 3. Individuals of the same sex but difference in gonads. They show high rate of fecundity. Various phenomena are there for reproduction. 1. Parthenogenesis. 2. Oviparity and viviparity. 3. Occurrence of generations in which sexes are unequally represented, males rare or even absent. They show high rate of fecundity, reproduction dependent on temperature and quality of host plant. Aphids are known for the polymorphism shown in different generations of the same species. 1. Different forms of the same sex either in the same or different generations. 2. Individuals with perfect or atrophied mouth parts. 3. Individuals of the same sex but difference in gonads. They show difference in their habits. 2. 1. Migration to different host plants. 2. Different modes of life of the same species on the same host plant. 3. Different habits of the individuals of the same generation. Life cycle. The following stages can be seen in the life history of an aphid. Eggs are laid by sexual oviparous females in the autumn. The egg stage continues throughout the winter. In the following spring the eggs hatch to form apteris, parthenogenetic viviparous females. These females give rise to many generations throughout summer of similar females on the primary host plants. Some of the progeny can be winged parthenogenetic females. The winged viviparous females migrate as swarms to secondary host plants and produce similar females. Towards the end of summer or autumn both eggs are laid by sexual oviparous females in the autumn. The egg stage continues throughout the winter. In the following spring the eggs hatch to form apteris, parthenogenetic viviparous females. These females give rise to many generations throughout summer of similar females on the primary host plants. Some of the progeny can be winged parthenogenetic females. The winged viviparous females migrate as swarms to secondary host plants and produce similar females. Towards the end of summer or autumn both apteris and parthenogenetic generations give rise to sexual males and females. The females are oviparous. The cycle repeats. Polymorphs in the life cycle of a migratory aphid. 1. Fundatrices, apteris, viviparous, parthenogenetic females which emerge in spring from the overwintering eggs. Sense organs, antennae and legs are not well developed, eyes are small. 2. Fundatrigenii, apteris, viviparous, parthenogenetic females which are progenies of fundatrices and live on the primary host. 3. Migrants, winged, viviparous, parthenogenetic females which develop on primary host and migrate to secondary host. 4. Alienicoli, winged and parthenogenetic viviparous females developing on the secondary host. 5. Sexuperae, winged parthenogenetic viviparous females developing on the secondary host migrate to the primary host. 6. Sexuales, form only once in the life cycle. Consists of sexual oviparous females and males which reproduce sexually to give rise to the next generation of fundatrices. Eggs winter apteris. Parthenogenetic. Viviparous. Fundatrices summer. Apteris. Parthenogenetic, viviparous. Fundatrigenii summer, primary host. Winged. Parthenogenetic, viviparous. Migrants summer, secondary host. Winged. Parthenogenetic, viviparous. Alienicoli summer, secondary host. Winged. Parthenogenetic, viviparous. Sexuperae end of summer, primary host. Sexual males and females, females oviparous. Sexuales winter, primary host, eggs give rise to fundatrices. Dipterans as vectors of diseases. Diptera is an important insect order as several species is known for medical, agricultural and veterinary significance. Most dipterans like mosquitoes and houseflies live closely to human settlement and cause severe problems including nuisance and transmit diseases to human beings. Mosquitoes as vectors. Mosquitoes can transmit diverse infectious pathogens and parasites that cause diseases such as dengue, Zika, chikungunya, West Nile fever, or malaria. In order to do so, though, a mosquito must bite a sick human first, thereby infecting itself with the pathogen. After some time, usually around 10 days, the mosquito itself becomes infectious. Mosquitoes can act as vectors for many disease-causing viruses and parasites. Viral diseases, such as yellow fever, dengue fever, and chikungunya are transmitted mostly by Aedes aegypti. This disease is spread through the bites of infected mosquitoes and cannot be spread person to person. The parasitic disease is collectively called malaria, caused by various species of Plasmodium is carried by female mosquitoes of the genus Anopheles. Lymphatic filariasis can be spread by a wide variety of mosquito species. Equine encephalitis viruses can be spread by mosquito vectors such as Aedes teinurensis. Tularemia, a bacterial disease caused by Francisella tularensis, as well as arbovirus infections such as West Nile virus is variously transmitted, including by Culex. Zika, though rarely deadly, causes fever, joint pain, rashes and conjunctivitis. The most serious consequence appears when the infected person is a pregnant woman, since during pregnancy this virus can originate a birth defect called microcephaly. St. Louis encephalitis, a mosquito-borne disease that is characterized by fever and headaches upon initial onset of infection, arises from mosquitoes who feed on birds who are infected with the illness, and can result in death. The most common vector of this disease is Culex pipiens, also known as the common house mosquito. Heartworm disease, a parasitic roundworm infection that affects dogs, is transmitted by mosquitoes. Adult heartworms infest the right heart and pulmonary artery, where they can cause serious complications including congestive heart failure. Mucidias vectors. The common housefly is one of the most widely distributed insects and has the ability to transmit disease to people. E.g., cholera, typhoid, shigellosis. This fly is commonly found in homes and restaurants. Because of its close association with people and its ability to transmit disease, it is considered a greater threat to human welfare than any other species of non-biting fly. Houseflies can fly for several kilometers from their breeding places, carrying a wide variety of organisms on their hairs, mouthparts, vomitus, and feces. Parasites carried include cysts of protozoa, e.g., Entamoeba histolytica, and eggs of helminths, e.g., Ascaris lumbricoides, Trichuris trichiora, and Enterobius vermicularis. Houseflies do not serve as a secondary host or act as a reservoir of any bacteria of medical or veterinary importance, but they do serve as mechanical vectors to over 100 pathogens, such as those causing typhoid, cholera, salmonellosis, bacillary dysentery, tuberculosis, anthrax, ophthalmia, and pyogenic cocci, making them especially problematic in hospitals and during outbreaks of certain diseases. Disease-causing organisms on the outer surface of the housefly may survive for a few hours, but those in the crop or gut can be viable for several days. Usually, too few bacteria are on the external surface of the houseflies, except perhaps for Shigella, to cause infection, so the main routes to human infection are through the housefly's regurgitation and defecation. A number of bacterial endosymbionts have however been detected, the greatest numbers being detected in the abdomen. Glossinidias vectors. The sisi are obligate parasites that live by feeding on the blood of vertebrate animals. Human African trypanosomiasis, also called sleeping sickness, is caused by Trypanosoma brucei. This disease is invariably fatal unless treated, but can almost always be cured with current medicines if the disease is diagnosed early enough. Sleeping sickness begins with a sisi bite leading to an inoculation in the subcutaneous tissue. The infection moves into the lymphatic system, leading to a characteristic swelling of the lymph glands. The infection progresses into the bloodstream and eventually crosses into the central nervous system and invades the brain, leading to extreme lethargy and eventually to death. Animal trypanosomiasis, also called nagana, when it occurs in bovine cattle or horses, or sura, when it occurs in domestic pigs, is caused by several trypanosome species. These diseases reduce the growth rate, milk productivity, and strength of farm animals, generally leading to the eventual death of the infected animals. The sisi fly vector ranges mostly in the central part of Africa. Dipterans as vectors of disease. It is an order comprising of terrestrial insects with great powers of leaping and running, except in certain acrididae. Flight is not well developed and alary organs are quite wanting in some species. Some families like Blattidae and Acrididae have aquatic species, but they don't have any special structural modifications. E.g., Blattidae, when submerged, keeps the abdominal apex in contact with the air. They are insects of comparatively large size. External anatomy. Head, hypognathous, rarely prognathous, frontoclypeal sutures are distinct, X-shaped tentorium without a central aperture. Framework of internal support within an arthropod head, formed by ingrowths of the exoskeleton, called apophyses, head widely attached to the thorax, free movement of head is limited. 1. Antennae, cetaceous, short or long. 2. Compound eyes, usually large and surrounded by band-like ocular sclerites, well-developed ocelli, usually 3 in number, absent in apteris forms. 3. Mouthparts, well-developed mandibles, maxillary palps are 5-segmented and labial palps are 3-segmented. 4. Thorax, prothorax is large, pronotum extended backwards, meso and metathorax together form a strong pterothorax. 5. Legs, unequal in length, hindlegs with enlarged femora due to deposition of strong tibial levator muscles and adapted for jumping except in family like Gryllatalpidae. Forelegs are modified into fossorial type with forotibia broad and toothed Gryllatalpidae. In Gryllidea, forotibia has proximally a pair of tympanal organs. 7. Wings. Fore and hind wings are unequal, forewings are small, sclerotized, leathery. In Tedygonidae and Gryllidea, forewings have stridulatory organs. Hindwings are membranous with large anal lobe folded fanwise 8. Abdomen, long and 11 segmented. Acrididae has a pair of membranous tympanal organs on either side of the abdomen. Cerci short and unsegmented except in Gryllidea. 9. Alimentary canal, straight or slightly convoluted, with large crop, gizzard-bearing teeth, midgut with anterior long cc. Malpighian tubules are numerous, forming an ampulla Tedygonidae, or opening into the gut Gryllidea. Hindgut has rectum with 6 rectal pads. Salivary glands are well developed with salivary reservoirs. 10. Nervous system. CNS has 4 to 6 abdominal ganglia. Sympathetic nervous system is well developed frontal and hypocerebral ganglia, a pair of esophageal nerves, corpora cardiaca, corpora allata, and a pair of ingluvial ganglia. Thoracic and abdominal nerves and neurohemal egans are well developed. 11. Tracheal system. Open type with 2 pairs of thoracic and 8 pairs of abdominal ganglia. Trachea has segmental air sacs. 12. Circulatory system. Heart elongated with 5 pairs of abdominal ostia and 2 to 3 pairs of thoracic ostia. 13. Reproductive system. Female genitalia are well developed. Ovipositor has 3 pairs of long valves. Male genitalia is well developed composed of a pair of styles on 9th abdominal segment. Male has a pair of multifollicular testes, a pair of vas deferentia anteriorly modified into epididymis, a median ejaculatory duct, a pair of seminal vesicles and a median mushroom gland. Female has a pair of penoistic ovaries, a pair of oviducts open into a vagina, a spermatheca, a pair of accessory glands and a genital chamber. The clitorial glands are well developed in families like Bladdedawick form highly developed ootheca. 14. Development. Eggs are elongate, laid singly or in clusters in or on the ground. The first instar nymph is vermiform prenymph. Total number of nymphal instars vary from 4 to 15. In apteris forms, metamorphosis is slight. Classification. Orthoptera has 2 suborders. 1. Encephera pterygonioidea, antennae longer than the body, tympanal organs on foratibiae, stridulatory organs on tegmina, elongated ovipositor, long-horned grasshoppers, bush crickets, mole crickets. The suborder has 3 superfamilies. A. Grillocridoidea, without stridulatory organs and tympanal organs but with long ovipositor. The families are Stenopelmatidae, Grillocrididae, Schizodactylidae, Rhaphidophoridae. B. Pterygonioidea, long antennae, stridulatory and tympanal organs, long ovipositor, winged and wingless forms are seen. The families are Pterygonidae, Brophalongosidae. Circa. Grilloidea, males have stridulatory organs, 3 segmented tarsi, ovipositor needle-shaped. The families are Grillidae and Grillotalpidae. 2. Calyphera, Acridoidea, short-horned grasshoppers, locusts etc. Antennae shorter than the body, tympanal organs on the first abdominal segment, stridulatory organs on femur, ovipositor short or absent. The suborder has 2 superfamilies. A. Acridoidea, 3 segmented tarsi, well-developed ovipositor. Family Acrididae. B. Tridactyloidea, segmented vestigial tarsi, ovipositor absent. Family Tridactylidae and Cylindrochetidae. Family Pterygonidae, long-horned grasshoppers. Antennae are filiform, 30 or more segmented, extending far beyond the apex of the abdomen. Tegmina, leathery forewing slope obliquely backwards, membranous, left one overlapping the right and shorter than the wings. Stridulating organ found on the left tegmen. Right tegmen has a smooth transparent area of tense membrane which acts as a resonator for the tegmina. They make loud sound at night. A pair of auditory organs are present on each foratibia. Winged Pterygonidae are green and live among foliage. Wingless forms are gray or brown in color and live in burrows, hollow trees, beneath stones etc. Ovipositor is longer than the body and used to lay eggs in the earth or in plant tissues in longitudinal rows. Eggs are not laid in ootheca. 5 to 6 molts are common in the family. Family Tetragidae, groundhoppers, pygmy grasshoppers. They are less than 20 millimeters in length with a long pronotum, which extends over the length of the abdomen sometimes to the tip of the wing. The first thoracic sternite is modified into a collar-like structure called sternomentum. They have scaly forewings and well-developed hind wings. Front and middle tarsi with 2 segments, hind front and middle tarsi with 2 segments, hind tarsi with 3 segments. Auditory and stridulatory organs are absent. They show sexual dimorphism both in size and color. They have hemimetabolous development, in which eggs hatch into nymphs. Family Acrididae, locusts and short-horned grasshoppers. Antennae are shorter than the body and may be filiform or clubbed. There is a ridge formed of about 80 to 90 pegs on the inner surface of the hind femora which forms the stridulatory apparatus which is rubbed against the tough veins on the folded tegmina. The auditory organs are present on either side of the basal segment of the abdomen. Ovipositor is inconspicuous and has short valves with which they excavate a hole in the ground in which 80 to 100 eggs are laid in a mass. A gelatinous substance hardens to form a tough covering for the eggs. There will be 5 to 8 ecdyses in the life history and 2 generations can be seen in a year. They are voracious feeders of vegetation during their nymphal and adult stages. Locusts. Locusts belong to the subfamily Acridiinae, characterized by the presence of a long spine on the prosterna in between the anterior legs. The genus Locusta includes the world's oldest migratory locusts. It has two species, L. migratoria and L. pardalina. They form large swarms when the population of these locusts reach the peak. They migrate from their breeding ground, the exact reason of which is not known. The locust migration is not driven by hunger since during this period, they don't eat much but depend on their fat reserves. Cessation of flight is said to be due to internal physiological causes like maturation of gonads and connected to the discovery of suitable breeding grounds. The resulting progeny gives rise to Danica phase, which is not gregarious but aids in the distribution of the species. This phase in turn gives rise to migratory 1. Schistosera gregea is an example of locust that caused vast damage to vegetation in northern Africa, Iran, Afghanistan and northern India. There are two phases, a destructive migratory phase, Phasis gregaria, and a harmless solitary phase, Phasis solitaria. Two phases are connected by intermediates called Phasis transcens 1. Phasis gregaria. The nymphs are black, yellow or orange in color. On attaining maturity, the color changes especially in males. The adults have concave pronotum, prominently constricted. The wings have greater length than solitaria. Hind femur is shorter than forewing. Body temperature is high. Color change is shown after sexual maturity. Both adults and nymphs are gregarious. They are diapause 2. Phasis solitaria. Nymphs are extremely plastic in their coloration, changing the color according to the environment. The imago has arched pronotum without constriction. There is no change in the color of male and female. Hind femur is longer than forewing. Body temperature is low. They always behave as solitary grasshoppers. 3. Phasis transcens. Based on whether developing towards solitary or gregarious phase, they show tendencies of developing the characteristics of both gregaria and solitaria. The gregaria phase develops only in specific breeding areas, where a particular combination of ecological conditions prevail. When the conditions are favorable, they form dense swarms, migrate to new breeding grounds, the resulting progeny develops into solitary or transcens. Locust breeding and life history. Breeding of locusts koiniki with rainfall and eggs are laid in moist soil in the transitional areas between desert and semi-desert regions which provide biotic resistance to them. Life cycle has 3 stages: eggs, hopper and adult. Eggs are laid in batches of 150 or more in the burrow of 8 to 15 cm depth in sandy soil. Depending on the temperature, eggs hatch in 11 halves to 4 weeks to prenymph and after a few molts become hoppers. Structure and behavior in the hoppers changes to scattered or concentrated manner depending on whether they enter solitary or gregarious phase. After 3 to 10 weeks, final molt brings in the adult stage which survive for 10 to 15 days. There are 2 breeding seasons during a year, spring and summer, monsoon depending on whether the rainfall occurs in spring or summer in different breeding homes of the locusts. Migration of swarms. 1. Eastward swarms, from the winter rainfall areas of Middle East Baluchistan to summer rainfall areas like Sindh, Punjab, Rajasthan, Uttar Pradesh etc. during May to July. In the wake of monsoon rainfall, these swarms lay eggs and breed here till September-November if the rains continue and temperature is high. 2. Westward swarms. From September, many new swarms from monsoon breeding areas fly westwards into the spring breeding areas of Baluchistan, Iran, eastern Arabia where they breed again in the following spring. 3. Southward swarm. From Rajasthan to east and south damaging the crops of vast areas of India but without breeding, they die off. 4. Northward swarm. Overwinter in North India where they breed and damage crops of vast areas of India. Swarms stop only when the climatic conditions become unfavorable either in monsoon or spring in the breeding homes, else they multiply from season to season, year to year. Till favorable condition returns they remain solitary. Each swarm can contain a single mass of about 40 miles in length and several feet in height in which there will be 2,500 individuals. Swarms can spread to 2.5 sqkm with 100 million locusts which consume 300 tons of food while they settle. Biotic Theory of Periodicity of Locust Cycles Prodden, 1969. This explains probable causes of phase, transformation and swarm outbreaks in periodic and cyclic manner. Drastic climatic changes in the breeding areas are solely responsible for the periodic locust outbreaks, causing transformation of locusts from solitary to gregarious forms. It is said that when climate changes, natural enemies of locusts are reduced causing their population explosion. They start laying eggs extensively, absorb more solar heat to produce melanin in the integument and wing. Then for searching suitable environmental conditions, they collectively swarm and migrate to long distances. In normal climatic conditions, conversion of locusts from solitary to gregarious as well as swarming and distant migration never occurs. Locust control is done internationally by various organizations like Locust Warning Organization, India, 1939 International Locust Research Center. Anti-locust operations under FAO. These organizations alert people about new outbreaks, swarms and suggest package schedule of locust control methods. Family Grillidae, crickets. They resemble Tetigonidae in having long filiform antennae, exserted ovipositor, stridulatory organs on tegmina and auditory organs on tibia. They resemble Acrididae in having 3 jointed tarsi. Tegmina are folded flat over the abdomen, right tegmen overlying the left, bent abruptly downwards along the sides of the abdomen. The stridulatory organ comprises of a file in a hardened area called scraper on the tegmina against which the file of the other tegmen works. There is also a vibratory area or tympanum on both tegmina. During stridulation, the tegmina are elevated to 45° with respect to the abdomen, and moved back and forth so that the file of one tegmen is rubbed against the scraper of the other tegmen. This movement in turn vibrates the tympana, which leads to the production of an audible sound. The cerci are exceptionally long and unsegmented, anal styli are short. Eggs are laid in the ground or in the pith of the twigs. The life cycle has 5 or more ecdyses. They are omnivorous, frequent hot dry areas and live in holes or burrows. Family Gryllothalpidae, mole crickets. They are subterranean in habit and seldom seen. The body is cylindrical in shape and covered by fine dense hairs. The head, prothorax and forelimbs are highly sclerotized, abdomen is rather soft. The forelegs are greatly inflated and modified for fossorial habit. The head bears thread-like antennae, compound eyes are much reduced. The 2 pairs of wings are folded flat on the abdomen, the hind wings are membranous, exceeding the tip of the abdomen in many species. In some the wings are reduced. There is no visible ovipositor. The females burrow into the soil to a depth of 30 centimeters and lays a clutch of 25 to 60 eggs. Females remain in an adjoining chamber to guard the clutch. Eggs hatch in a few weeks and nymphs are voracious feeders of plant material. The nymphs resemble the adults apart from the absence of genitalia and wings.